Hey, good morning. Why don't we go ahead and get started? And if folks are trickling in, they'll join us. My name is Letitia Stein. I am the health editor at the Philadelphia Inquirer. So I oversee a team of reporters that covers everything from measles risk to opioid addiction to the finance of health systems, changes in Medicaid. We also cover a region with a high concentration of medical institutions that have a storied history of so-called firsts. The nation's oldest hospital, the original polio vaccine, in more recent years, advances that we now know as CAR T therapy and the mRNA technology that powered the first COVID-19 vaccines are all stories that essentially began in Philadelphia. So last year on May 9th, an email landed in my inbox announcing an embargoed medical journal report on the quote unquote world's first patient treated with personalized CRISPR gene editing therapy. Candidly, I almost mistook it for one of the superlative pitches that I get all day long from PR outfits. But thankfully, I didn't push delete because as I read more closely, there was a lot here that looked as though it could be for real. The New England Journal of Medicine was about to publish a study. The work had taken place at the Children's Hospital of Philadelphia, which is preeminent nationally. I knew at a minimum that we were dealing with a nationally competitive story that would require specialized and careful reporting. Every health training I have attended in a decade plus as a health journalist has warned me to be careful of phrases like world's first and historic breakthrough, right? This is the kind of language that rarely holds up on closer inspection. Today, we are fortunate to be together with some of the people who are in this space to help us unpack what is, in fact, a one in a million case and success story. I am grateful that AHCJ leaders have given us an opportunity to include some remote speakers, which I'll bring online now. These are the people who have been immersed in this world for the last year. So just one moment. So thank you. So the format of what we're going to do today is just a little bit different than a traditional panel. We're going to start by hearing from Dr. Kiran Musunuru, one of the physician leaders involved in this, and Nicole Muldoon, whose newborn son, KJ, received this life-saving treatment. They are joining us via Zoom. Then to keep things personal for those in the room, we're going to switch to more of a fireside chat format between myself and Sara Whitlock, who's an expert in science communications. And we'll talk a little bit about trying to cover breaking news around research, best practices, some of the challenges that were unique to this story. Um, and we will also, of course, make sure to have time at the end for questions for all. To keep things lively as we're going through our conversation too with Nicole and Kiran, I'll occasionally stop and play a couple videos so we can see as much as was available to be seen of what was playing out last year at the Children's Hospital of Philadelphia at CHOP. So with that, I'm going to kick us off with some introductions. Let's start with our remote panelists. Dr. Musunuru, Kiran, and Nicole, can you all please introduce yourselves? And then Sarah will do the same.
Certainly. I'm Kiran Musunuru. I'm a professor of cardiovascular medicine, genetics, and pediatrics at the Perelman School of Medicine at the University of Pennsylvania in Philadelphia. I also have an appointment at the Children's Hospital of Philadelphia, which of course is right across the street from the university. And that fancy sounding title with a lot of different departmental affiliations means I work in different sandboxes, but I think what's important to know is as a clinician, as a practitioner, I'm an adult cardiologist, which means I take care of typically more elderly patients who have heart disease, heart attacks, heart failure, rhythm issues, and so forth. Those are my typical patient panel, which I think is perhaps relevant today, as we'll get into. But I do cross over through my work in genetics. I have a research laboratory at the University of Pennsylvania, and that's where I spend most of my effort. And more recently, I've been almost entirely focused on making so-called gene editing therapies for a whole variety of patients, those from who are having heart attacks along the lines of what the patients I take care of in my clinical practice, but then more recently extending it to the other end of the spectrum, rare diseases, ultra rare diseases, what we would call N of one scenarios, all the way to the time of birth, as you'll hear about today.
Nicole?
Hi, everyone. My name is Nicole Muldoon. Professionally, my background is in education, but more importantly, I am a mom of four kids, one of which being KJ, that we're going to talk about today. And the newest hat I have been wearing is also in patient advocacy.
Thank you both. Sara, can you introduce yourself?
Yeah, hello everyone. My name is Sara Whitlock. I am based at, I think so. Can everyone hear me? I'm Sara Whitlock. I'm based at the American Association for the Advancement of Science at a group called SciLine. So we are a group that focuses on connecting reporters with scientist sources for their stories. Professionally, my background is first in science and then in science writing. So I did a master's degree before switching gears, doing a lot of freelancing, and then coming to SciLine to kind of help people connect with reporters. I've also led our vaccine response this past year. So I've worked on developing resources for reporters covering vaccines throughout a pretty tumultuous year.
Great. Well, thank you all so much. And also, anyone in the room, if there are any technical pieces that you're struggling with, please just pull up your hand, let us know. We'll get it sorted out. So let's just begin with where the story begins, right? With a patient, a child now known worldwide as baby KJ, who had a rare disease. And in fact, the New England Journal study that came out last year was titled as followed: Patient-specific in vivo gene editing to treat a rare genetic disease. Dr. Musunuru, can you start by explaining to us a little bit what baby KJ was facing and why a novel form of gene editing was considered in this case?
Certainly. So we all need protein, right? We all eat lots of protein in our diet. We need it to maintain healthy bodies. When we eat protein in the diet, it gets broken down into different parts, a lot of which are useful to us, nutrients that we use to keep ourselves healthy. But there are some waste products as well. And so one of those waste products is ammonia, which is toxic. You don't want it in your body. Now, the reason we're able to eat as much protein as we want, the vast majority of us are eating way more protein than we really need. It's just endemic to the American diet. And we don't get into trouble is because we have ways of handling things like ammonia. In the case of ammonia, each of our livers has a series of 6 enzymes. They all work as a single pathway. They start with ammonia and they turn that ammonia into something called urea, which is not toxic to the body and in fact is very easy to clear from the body. It's actually the primary component of urine. That's why it's called urine, because it has a high amount of urea. So that's why we can handle as much protein as we can. Now, those 6 enzymes in the liver, if any one of those 6 enzymes is broken, then the body can't process that ammonia and it just builds up in the body, builds up at toxic levels, can cause irreversible injury to the brain. And if it is a prolonged enough exposure to that high level, can cause a patient to go into a coma and then eventually death. And all it takes is any one of those 6 enzymes being faulty. Now, how does that happen? How can these enzymes or any one of these enzymes be faulty? Well, each of these enzymes is made by a gene in our DNA, the source code, if you will, the blueprint for each of those enzymes. And if there's a misspelling in any one of those 6 genes, then the corresponding enzyme that's made from that gene is going to be broken. And so if a child has inherited misspellings in any one of those 6 genes, they'll be in a situation where they're not making all 6 enzymes. As soon as they are born, as soon as they drink mother's milk for the first time, which is protein, right? They break down the protein. Ammonia results from that. They can't process the ammonia and it builds to a very high level. So within 24 to 48 hours, that kid becomes very, very sick. And if things are not done immediately, like dialysis, some pretty heroic things to clear the ammonia from the blood, it becomes such an issue that, again, you can go into coma and then, very quickly pass away. And so the mortality rate with these diseases is very high, in excess of 50%. If a kid is fortunate enough to be in a place where somebody recognizes what's going on, checks an ammonia level, realizes, hey, this is a problem, and can get some treatment in time, for example, dialysis to clean the ammonia out of the blood, then the kid can pull through. But that doesn't really fix the issue because anytime they eat protein, there's that risk that some ammonia is made and that ammonia is not cleared enough from the body, builds up to toxic levels and has irreversible injury to the brain. And so what can happen is kids can get hits over time and they have a decline in neurological function and brain function over time, and they decline over time over months to years. The only way we have of actually fixing this issue is to replace the entire liver, right? If you have a broken enzyme in the liver, and it sounds very crude, but that's the state of the art. You replace the entire liver, take a liver, a healthy liver from someone else. that has all six enzymes working, then you're treating the underlying issue. The unfortunate part is that a liver transplant brings a whole different host of issues. One, it's major surgery. So it has its own risks. And the younger a patient is when they have a liver transplant, the higher the risks of having a bad complication or even dying from the surgery, just because bad things can happen, obviously, during major surgery. Even if they pull through and the surgery goes fine and there aren't major complications, it's someone else's liver. It's not their own liver. And so they're at risk of their immune system rejecting that liver at any time. And so they have to be on pretty powerful immunosuppressive drugs to keep that liver in place. Um, and when you're on lifelong immunosuppression, that brings its own host of risks. Risk of infection, increased risk of cancer for the lifetime, can cause toxicity to the kidneys, and so forth. So you're really just trading one chronic disease to another chronic disease. A better situation for sure, but still, not full health. Um, the other dimension to this is that it's not always possible to get a liver transplant, right? So you need to have a match. It needs to be available, right? If you're getting a liver, then there has to be a donor, and you know the ability to get a donated liver is obviously it's a scarce resource. It's not always possible to get one when you need one. And so a kid who's born in this situation with one of these six enzymes broken, the technical term we use for these diseases are the urea cycle disorders. The urea cycle is that chain of six enzymes that together turn the ammonia into urea. So if you have any one of these urea cycle disorders, you're in the situation where you're at constant risk of the ammonia levels building too high and causing damage, causing injury. Even something as benign as a common cold or stomach flu, any common childhood illness can put stress on the body and cause the ammonia levels to surge. Accidentally eating too much protein in the diet can cause the same problem. So you have to heavily restrict the amount of protein in the diet. You have to give medications on a continuing basis to try to keep the ammonia levels under control. And despite all of that, can still occasionally slip and the ammonia level rises and it causes injury, and you get this decline over time. And then it just becomes a question of hoping that a liver transplant becomes available. So that was the situation that KJ was in when he was born, and Nicole will tell you more about that in a moment. But what we decided to try to do, and when I say we, it was myself, but certainly I would be very remiss if I didn't fully acknowledge my scientific partner in all of this, Rebecca Ahrens-Nicholas, who, like me, is a physician scientist. Like me, she has a research laboratory and studies diseases and gene editing therapies in her laboratory. Unlike me, she is not an adult cardiologist. She is actually a pediatrician who takes care of kids with urea cycle disorders. Unfortunately, she couldn't be with us today, so I'm deputized to be here on her behalf. But the team of the two of us decided, we let's try to do something different. If it's misspellings in the DNA in a gene that are causing the problem, what if we could use this new technology, gene editing, and actually be able to correct the misspellings at the source in the liver, basically fix that broken enzyme from within, from the inside. So instead of having to replace the whole liver, we could give a therapy that would deliver a gene editor into all the cells in the liver, fix enough of those misspellings, enough of those cells that there's enough enzyme now made to be able to handle that ammonia and turn it into urea and get the kid out of this tough spot. Nothing like that had ever been done before because there really was no playbook by which one could actually say, hey, this kid has a very unique misspelling. They may be the only person in the world who has that misspelling. We want to actually make a personalized therapy, a bespoke therapy that will do the gene editing, that will fix that specific misspelling to the correct spelling, and then be able to make that drug from scratch in a very short time. And, again, this is a situation where the clock is ticking. You don't have years to develop the drug like you normally would with traditional drug development. In this case, there's a kid and, at any moment their ammonia levels could surge. They could have that irreversible injury, that function they're never going to get back. And so the clock is ticking. You're really, trying to get this done in a matter of a few months rather than years and years. So those were the challenges we faced. And something like this, to our knowledge, had never really been done before, at least not successfully. But we committed ourselves to it. We started with the misspelling we knew KJ had because he had had his entire genome sequenced right after he was born and was diagnosed with his particular flavor of the disease. which I should mention is called CPS1 deficiency. It's one of the 6 or 7 urea cycle disorders. It just, the particular enzyme that was broken out of the 6 enzymes was CPS1, hence the name CPS1 deficiency. But the beauty of gene editing is it doesn't really matter which gene is broken because you can tell CRISPR where to go wherever the
Yeah.
misspelling is and actually fix that misspelling. And so mounted a 6-month campaign, brought in academic partners, industry partners, basically all the friends we had um, in our scientific community who we could recruit to the cause. And then in just somehow miraculously in 6 months, we're able to design the drug and then actually get it manufactured and then engage with the FDA and get sign off from the FDA to actually treat KJ with this batch of drug that was made
Yeah,
just
thank
for
you. Thank you. So we'll be exploring each of those steps more. Thank you. Before we get too in depth, Nicole, you just had a baby. This isn't a patient, right? This is a child. He's got 3 siblings at home. Talk to us about what you knew when and how the very early stages. What was the decision like for you here?
So at birth, KJ was 5 weeks premature. So we already, before we even knew he had the diagnosis of CPS1 or that he had this urea cycle disorder, we knew that he was a preemie, NICU stay might be in the works for us, and we were ready for that. They said it could be till he was of term age. But the night that he was born, he seemed to be doing really well. They were very happy with, how he was doing, and we're talking about moving him to out of the NICU to um, the regular nursery and then into my room. And then I guess my husband and I fell asleep, around 9:30, waiting for the baby to come down, and we woke up around 1, and he still wasn't there. So my husband went up to the NICU just to check, and he was back on a CPAP machine or a breathing machine to help give him support. And just weren't liking to how things were starting to look. He was not really wanting to eat. He was, pretty lethargic. So that's when they started to run some tests. And by the next day, um, I remember vividly my husband raising his arm in the air, and when he would let go, it wouldn't just drop down like a baby's would. It would shake down with this like rigid movement. And at the same time we were noticing that, a doctor came in to us and said, your child is very sick. We need to act right now. And lucky, we were at the University of Penn, I should mention. And lucky for you, we have the Children's Hospital right across the street. So they immediately brought him over. And from there, it was kind of a whirlwind. KJ went through the underground tunnels that run between University of Penn and the Children's Hospital, and went right up to the pediatric intensive care unit to start dialysis of the blood. And that's when we started genetic testing, all of these different things. And, we're getting all of this information thrown at us at the same time. And we're worried about this little 4-pound baby that is 5 weeks early and we thought was, just a preemie, but other than that, overall healthy. They did test his ammonia. Normal measures of ammonia in a, healthy person typically range between 9 and 33 is like the normal measurement. KJ's ammonia levels just at just over 24 hours old were just in the 1600s, almost 1700s. So a little 4-pound baby that was just born, this little preemie, with an ammonia that high was, pretty significant. And they were like, we need to act right now. So my husband and I, just dove right in. We decided that we were going to, give KJ all the support that we could, listen to the doctors, ask the questions, play devil's advocate. We just wanted all the information, regardless, good, bad, ugly, to make sure that we were making the best decisions on behalf of KJ, on behalf of his health, and to try to set him up for the best possible life that he could have. So when doctors would come in, we'd listen, entertain all of the ideas, all of the information they were giving, and then ask questions and do our research. There was a very short amount of time where it was like, we felt sorry for ourselves. There wasn't really none of that. We really just dove right in and wanted all armed ourselves with as much information as we possibly could in order to make sure that our baby would be here with us.
Kiran, you're talking about bringing gene editing to literally a 1 in a million case, but the therapy that we're discussing here clearly is being designed and considered in a wider context. So before we start talking very specifically about baby KJ, can you tell us a bit about gene editing? What's the promise? What's been tried? Where is the state of this technology?
Yeah, so the standard gene editor, if you will, which is also known as CRISPR, a term I'm sure most of you, if not all of you, are familiar with. It's a two-component system. One component is the protein that you can think of as the editor. It's what actually makes changes to DNA. The second component is a so-called guide RNA molecule. You can think of it as a molecular GPS. That protein and that GPS come together to form the molecular machine. If you can deliver that machine into the cells in the appropriate part of the body, what it will do once it's in the cells, that CRISPR machine will scan through all the DNA in that cell. We're talking 46 chromosomes. We're talking about 6.1, 6.2 billion letters in total. And the GPS, that second component, has within it a 20-letter sequence. Whatever you've programmed into that 20-letter sequence, the machine will find somewhere in those 6 billion letters with pinpoint accuracy. The machine will find that spot, engage there, and then make the change, right? And so the beauty of the technology is you can repurpose it over and over and over again to disease A, to disease B, to disease C, all the way to diseases X, Y, Z, simply by changing that GPS address, pointing it from one gene to another gene. And so where this CRISPR gene editing first got traction was, believe it or not, treating various types of heart disease. And the idea there was to point CRISPR using that GPS address to a gene that was driving heart disease. And it might be a protein that leads to heart failure. It might be a gene that brings up cholesterol levels in the blood. And the idea is that you could use CRISPR to turn off the offending gene in the liver. And in doing so, basically treat the disease at its root cause. And because you're doing it at the DNA level in a gene, in the source code, once you've made the change, in principle, it's durable for a very, very long time, really for the lifetime. And so I assume you're all medical reporters, otherwise you wouldn't be there in the room. But you may have heard just this past Monday, a few days ago, an announcement from Eli Lilly, world's largest pharma company, about the results of a clinical trial that they've been running. And this was published in the New England Journal of Medicine on Monday as well. A trial of a gene editing drug using CRISPR, turning off a cholesterol gene called PCSK9 in the liver, results from about 35 patients, showing that it works really well. You can take this drug, the GPS is tuned, it has that 20-letter address that tells it, hey, go after this cholesterol gene, turn off the cholesterol gene. And when patients with heart disease and with high cholesterol levels were given this treatment, it worked really well. Turned off the cholesterol gene, their cholesterol levels plummeted 40, 50, 60%. And in the trial so far, that's been durable for more than a year and a half in the patients who've received it, who've had the most time since treatment. And the expectation is it will be good for the lifetime. So we're seeing this play out with common diseases, with things like heart disease, which is the leading cause of death worldwide. But the beauty of this technology is that all you need to do is change that GPS. Instead of turning off a cholesterol gene, which could be helpful ultimately for millions of people, you could target it so that, change the address so that it fixes a misspelling in a patient who may be the only person in the world who has that particular flavor of disease, who has that particular misspelling, and really make a personalized bespoke therapy. And the beauty is that it's essentially the same drug. So once you've gotten the experience and understand how that drugs work and what its safety profile is, and have data from dozens or even hundreds of patients, it's very easy to repurpose it and then start treating entirely different diseases. And that was the operating premise here.
Let's start talking a bit about the speed and the timing here. From birth to infusion, I believe we're talking about a period of 6 months. What made it possible to move so fast, both from a technological standpoint and the regulatory process? Kiran, could you start talking about, what research and science was involved in reaching this point? And then Nicole, can you talk to us about what this was all like to experience at the bedside?
Yeah, so again, it comes down to the unique nature of this drug, the idea that you can reprogram it, that you can just change this GPS address and redirect it from one disease to another. So, I mentioned this drug that Eli Lilly has in clinical trials. What I failed to mention, I should have mentioned, is the work behind that therapy actually originated in my laboratory going back more than 12 years now. And so I had a lot of familiarity with this kind of drug and how to make the drug. And so in KJ's case, we did not have to start from scratch. There was already a drug in clinical trials that had already shown that it could work quite well and that it was reasonably safe. I was able to take that drug that I'd, helped to develop over the past decade, and just change that GPS address so that instead of turning off a cholesterol gene, it would fix KJ's specific misspelling. And I don't want to trivialize what we did because it was a lot of work, but fundamentally, in concept, it was just changing 20 letters in the GPS. And because there was already a track record with this kind of drug, because it had already been in patients, very different patients, older patients with heart disease, but it already had a track record. It already had safety data. It became much easier for Becca and me in developing a different flavor of this drug, same drug, but just a different customized flavor of this drug, to go to the FDA and say, hey, you've already seen this drug. You already know this drug because you've seen it in clinical trials from Lilly or this other company or yet another company who are making these types of therapies. We're not starting from scratch. All we need to do is show you that, look, this version of the drug is well suited to correct KJ's misspelling. If we can show you that and give you a little bit of extra data to show that it's safe and so forth, can we rely on the track record that has already been generated for this kind of drug and that you, as the FDA, already have full access to in a way that no one else does, right? Because they see everything. Would that reassure you that it's reasonable for us to go ahead and do what's called single patient expanded access IND application? It's often called compassionate use. Some people call it right to try, but the formal term is single patient expanded access IND. Would this be acceptable to you? And they were very reasonable and understood, our argument and agreed. And so they committed that when the time came and the drug was fully developed, which ended up happening about 6 months later, that they would very, very quickly review it. It only ended up taking them a week, which is, lightning speed for the FDA, and then gave us the go-ahead to treat KJ.
Nicole, how was all of this experienced by you and your family and your...
Yeah, so my husband and I are opposite thinkers, I'll say, and Kiran may agree. My husband's a much more emotional thinker, whereas I'm very much more analytical. I need to know like all of the information before I agree to anything. Whereas my husband's very much a gut guy. So initially, when Dr. Rebecca had come to us, I wasn't at bedside, my husband was, and she came with him about this idea or this research and asked if we could participate, never really knowing or thinking that it would be ready in time for KJ. But we agreed because we knew what it felt like to have a baby be so sick. And we wanted to participate in the research to help in any way that we possibly could other families that were going through this, even if it wasn't going to be available to help KJ, hoping to be able to alleviate some of that stress, illness, whatever we could for other families. So that's kind of how we approached the initial agreement to participating in the research. And then, a couple weeks would go by, and then Dr. Rebecca would come back and say, hey, things are moving, along. Are you guys still good to participate? Are you still comfortable? And we'd have some questions we'd ask. And I'll never forget, it was I believe in January, like right after the holidays, Dr. Rebecca came to us and said, you know, we are at a place in research in our what we've been doing in the lab that we think this may actually be able to be trialed with KJ. Are you guys still comfortable? And we said, absolutely. At this point, KJ had already had a few bouts of hyperammonemia. So he had a few episodes where he got really, really sick again. So at this point, we had seen him, fight through severe illness and seen him struggle and seen him go through things that you would hope to never see anybody go through, let alone your newborn. So when she came to us, it was like the light at the end of the tunnel. And our kids at home understood, that KJ was sick. I don't know if you can hear him in the background. KJ was sick, but he wasn't contagious. We incorporated them as much as we could into the hospital setting. And that KJ would live at the hospital until we found a way to make him better in order to come home, whether that was transplant. And then thank God, thankfully for Dr. Kiran and Dr. Rebecca, this gene editing idea, whatever, was going to be the best course of action for him. So it was very much a collaborative effort in just hoping and praying and, putting all of our trust and support in Dr. Kiran and Dr. Rebecca to, have this happen. And then when the actual day came, all of the adults in the room were a nervous wreck and, counting down the minutes that were clicking on the clock, and KJ slept through the entire thing like it was no big deal. And he was the one that was getting this first time ever done treatment that hopefully would help, make his severe and so rare disease even just a little bit more manageable. So it was kind of a whirlwind of a 6 months, but I would do the 6 months over again for the same outcome. I'll say that.
Let's talk about that outcome. How did you know this was working? And what was that trajectory like in those initial weeks and months?
Kiran, do you want to take that first from the medical standpoint?
Yeah, I can take that. So within the next few days after KJ received that first treatment, it wasn't immediately clear whether it was going to work, right? These things can take some time to unfold, but there were some promising signs. His ammonia levels looked very good. Things looked very stable. There was just a sense that maybe he was a little better from a medical perspective. Enough so that we realized the only way we would know for sure is if we challenged him with extra protein, right? So that's the key to this whole thing, as I explained earlier. You eat protein, you break it down. Ammonia is one of the waste products. In a kid like KJ, before he was treated, it was dangerous, and he couldn't tolerate much protein. He was on a heavily restricted diet, and because of that, he was tiny for his age. I think the 8th or 9th percentile body weight for his age, right before we treated him. And so he said, what if we give him a little more protein? How will he do? If nothing happened, if this didn't work, then his body will very quickly tell us this was not a good thing to do because he'll start making more ammonia, he'll start building up, and then we know to back off. So brought it up 10%, and then the next day measured the ammonia level. And so I was actually out of country at the time. So a few days after KJ got his treatment, I was attending a scientific conference, a gene editing conference, as it happens in Ireland. And so Becca would get the information in what for her was the early morning, but because of the time zone difference for me, it was the late morning. So I'd be on tenterhooks like all morning long, waiting for that text from Becca. What is the result? What's the ammonia level looking like? And so as each day went by, protein brought up and the ammonia level remained stone cold normal. And then the next day, another 10% up. Once again, ammonia level stone cold normal. And so this transpired over several days, and it wasn't like there was a eureka moment, but each day just a little bit more confidence, a little more hope, a little more being convinced, hey, maybe something actually happened here. Maybe this has actually helped him. And then before we knew it, a whole week was out, and then he was actually getting more protein than is recommended for his age. And again, his ammonia level was staying, quite stable. So that was the first sign, at least for me, that wow, it does seem like something has changed. It does seem something is different, and he's tolerating a normal amount of protein in his diet for someone his age, for the first time in his life.
Nicole, what were you seeing?
I would agree. I definitely my husband and I were very keen on waking up first thing in the morning and just checking the labs that we knew he had gotten done the night overnight. So we were, routinely checking the labs as well. And we had got to a place where we knew what was normal, abnormal in the levels and the numbers that were showing. And there was a consistent level of normalcy, especially with the ammonia and the glutamine levels. And for those that don't know, glutamine is a marker for ammonia in the blood. So if the glutamine starts to rise, then the trajectory of the ammonia rising, they kind of go hand in hand. So we started to notice the same thing, but just from appearance wise, I started to notice he was getting a little meat on his bones. His coloring went from more of like pale, very, very pale or almost gray to a little bit more pink in his cheeks, and just activity levels. He was more interested in playing for longer periods of time and, more active and alert. He was always very happy, very smiley, but he was definitely seemed to be more engaged in wanting to play too. So that energy level was there too. So, everybody was thinking to themselves, like, we think something might be working here. But we weren't saying it out loud at this time after the first dose.
So we have seen the FDA come out with two, as I recall, two announcements related to different pathways based on this baby KJ gene editing therapy. Kiran, can you talk to us about what the conversation is, how it's unfolding? And I believe we've even had action within the last month, right before the FDA commissioner left. So can you bring us up to speed?
Yeah, so the FDA, I should start out saying, at least from my perspective, they have been wonderful partners. I know there's been a lot of controversy over the last few months, in part because of the changes in leadership, and in part because of some controversial decisions made by the FDA with respect to certain drugs and approvals versus non-approvals. But my personal experience, Becca and myself, as we've engaged with the FDA extensively over the last 2 years, it has been a wonderful experience. They've been terrific partners, keeping us on our toes. As recently as actually literally just a few minutes ago, I got a text saying the FDA wants to meet with us today. I'm not sure about what, but when they say we want to meet, it's like, okay, we're going to put everything else aside and meet. But it's been a great experience. And the substance of our conversation with them is, how do we go from a single kid like KJ? How can we scale this up and get this kind of therapy to all the patients who could benefit from it? Right? Because again, it's intrinsic in the nature of the technology. If, as I think we've shown, you can take CRISPR gene editing and just change that GPS address suited to one person's misspelling and actually make the drug relatively quickly and give it to that patient and actually have some improvement in their condition. Well, you'd want to be able to do that over and over again, not just for any one patient, not just for any one disease like CPS1 deficiency, not for any one group of diseases like the urea cycle disorders, all six of those enzymes, but really, if it's the same drug you can use over and over again, and there's some fix of a misspelling in some gene in the liver that, if you could correct that misspelling, would lead to better health, regardless of what the label we put on the disease name or whatnot. Why shouldn't we be able to do that now that we know how to do this? How do we go from one patient to being able to scale this up to approved therapies? And it's really important for us to be able to get to approved therapies, because Once you have the approval, then you have a fighting shot at getting reimbursement from insurance companies, from payers, and so forth. And none of this is economically viable until you actually get to the point where you can get reimbursed for these therapies. And so we've been working very hard with the FDA, having many meetings with them, to get a clinical trial up and running, a very unusual, maybe unprecedented sort of clinical trial, what we call an umbrella of umbrella clinical trial. And what I mean by that is that we want to be able to recruit any patient like KJ who was born with a misspelling in any one of 6 or 7 different genes that can cause these urea cycle disorders. It doesn't matter exactly which misspelling it is. The idea is that we can use CRISPR to fix that misspelling. So any patient with any of these 6 or 7 diseases, with any misspelling we think we can fix with CRISPR, we want to be able to enroll them in the clinical trial, be able to make a bespoke version of the therapy that we gave to KJ, for that particular person who's enrolled in the trial, and be able to treat them. And we want to be able to do this with full FDA oversight. We want to do it in a rigorous way where it's in the context of a clinical trial where we're gathering data and we're being very intentional about how we design the trial and gather the data, because we want to get to an approval at the end of the day. And we don't want to have to wait, many, many, many years. We want to get to approval sooner rather than later, like 2 to 3 years would be wonderful. So we've been working on our end to get this up and running. On the FDA side, they've rallied to this concept. I think, they're human beings too, just like the rest of us, and they hear a story like KJ's and they get some inspiration from it. And so both at the leadership level at the FDA, notwithstanding the rapid changes in the leadership recently, but also the career staff, the people who are actually doing the day-to-day work and who have been there for many, many years, even decades in some cases, have rallied to this. And, just a few months ago, the FDA announced a new, what they call plausible mechanism framework. And, without getting into the details of exactly what those specific words mean, a path that allows us to get to this sort of platform of individualized, personalized gene editing therapies, a path to getting that kind of platform approved much more quickly than has ever been possible before. So it's been really gratifying to see that sort of response from the FDA, and we're hoping to be among the first to be able to use that new framework to get these kinds of therapies to the patients who need them.
So KJ, I think, was named one of the most influential persons of 2025, 2026 by Science. Dr. Kiran and his partner, Rebecca, oh, here he is. They've been recognized by Time Magazine. Lots of accolades for this work already pouring in. Nicole, talk to us some more about what's going on, and please bring KJ back if he's available.
Say hi. Say good morning.
Hey, KJ.
Dr. Kiran, say hi.
Hey, KJ.
So KJ is doing fantastic. He's walking, he's running, he's keeping up with his 3 older siblings. My older children are 15, 5, and 4. So KJ has a lot of keeping up to do with them. He loves sports. He loves the Phillies and the Eagles. He can throw a ball across the room. He can hit a ball off the tee with, better than some 3-year-olds that I know. And all of these things, even small milestones like starting to talk, these are things that when we initially got diagnosis, we didn't know if he was going to see the next day, let alone 1 or coming up on 2. So seeing him meet even little milestones that as a parent, you write in a baby book and then forget about, are, even more important for him. He's very, very active. Say good morning. He likes to eat. He has a very healthy diet. He's still on a somewhat of a restricted diet with his protein, but way more than he could have received previously. You see Dr. Kiran, I know that's your buddy. Say hi. But he's doing really well. Hi, Kiran. Sit down. So we're just happy that, this opportunity, we were at the right place at the right time. And, Dr. Kiran came across KJ and you see Kiran, and asked, if we would like to participate. That will be a decision that I will never forget making. I'll never regret making. And now we're partnering with Kiran and Becca to, help advocate for other families like KJ so that, when you think of rare diseases, there could be a handful of kids that have, the same as KJ. But when it comes to rare disease, they're throughout the world. It's not so rare. There's millions of them. So we don't want to make, this more available, readily available for other families, so they don't have to experience what we experienced before gene editing. That's kind of our mission now.
Thank you. Thank you for sharing that. Let me bring us to the room for a few minutes, and then we're going to switch to questions so that you have a chance to talk some more. And it's perfectly okay if you're distracted by the video. About what this story was like and switch here, I mean, which was like to cover, and what, in hindsight, it's quite clear. This is a big deal, right? At the moment that this was all landing, I am a health editor at a newspaper or news outlet that is still fortunate to have a robust health team. However, my longtime science and medical reporter just left. I was faced with, needing to get up to speed on multiple things that you don't cover all the time, right? From gene editing to rare diseases, to, you name it. There was a limited amount of time. We also had limited access to the sources involved. I think there was one press conference scheduled with the doctors and with the representative of the Muldoon family. So, Sara, what is one supposed to do this right?
I guess so. When you're faced with one of these situations where you get a press release, it says a lot of things like you were mentioning that might seem kind of hard to believe. This is a breakthrough. This is a first. I think a couple things that I would recommend and we would recommend as SciLine, as in a position of helping people get up to speed on their reporting, is to first maybe ask a couple of questions to stress test that first. So you might ask yourself questions like, was this technology possible 5 years ago? And that's really going to help you differentiate between what is the institution, doing their best to promote really good work, framing this as, versus what is actually the reality of the situation, what led us up to this place. And I think, for example, in this situation, we can easily see that, no, this probably wasn't possible 5 years ago. This really is a major milestone and a big breakthrough. Another good question as you start interviewing these sources would be to ask something like, what would have made this a failure? What would have caused this to not work in your mind? And I like this question a lot because it gives the people you're talking to an opportunity to define their criteria for success. And as you continue to ask this question over and over again, you will get to see whether those criteria are shifting over time. And that may or may not be a bad thing, but it is an important thing to be keeping in mind. So those are a few first points that I can start with.
To this day, I still feel totally comfortable. And well, tell me if I'm wrong about this, with world's first language, though no reason to believe it's not. But how do we prove perfectly what is and isn't the world's first? I think I've been using phrase like first of its kind. I think that we certainly we can call this, significant advanced landmark therapy. It is I'm trying to break through words. Sorry. Oh, is this better? You hear me now? Is this better? Okay. I was going to say, some of the language around this it's tricky, right? I prefer first of its kind, a world's first personally, although you can tell me, if you think that's wrong. I think at this point we can talk about, landmark therapy and breakthrough therapy. But the language was hard here. It was certainly important for us to reach out to some independent experts as much as we could, to try to, get a sense of, hey, a New England Journal article, right? Like, does this look like this is indeed, kind of something that hasn't been done before? Significant advance? The answer was yes. Extremely promising. One thing I didn't quite realized to do at the time was to make sure that we were reading all the companion pieces in the journal. There were some notable experts commenting on this that would have been helpful for us to have. What are some of the other things you can do when you're reporting on deadline? And, you can push things so far, but also you need to be very kind of thoughtful about your word choices and your expert choices.
So you're asking what are other
So, find the independent expert, what are kind of like the basic steps?
Yeah. I mean, I think you've highlighted a lot of the really excellent ones in terms of finding that independent voice early on is super important. Sometimes that can be a real challenge on deadline, especially as you mentioned, you had lost the reporter who might have been closest to this beat right in that moment. So finding that independent voice, I can maybe give some tips on how you might go about finding these people. So one of the things that I'm doing every day is helping people connect with a scientific source that's like exactly right for their story. So things that I would recommend when you're looking for that independent voice would be to do things like, you're looking at their tape to see what kind of good communicator they are. I would recommend also looking at their academic conference presentations, even though they're going to be a little technical. You can often tell a lot about what kind of communicator a person is going to be because they're going to have to take a step back for their colleagues. I would also recommend, as you're trying to frame this and you're trying to find someone who can help you understand the level of breakthrough, to be not forgetting about teaching faculty at schools that may not have a big research program. If they've done their research in their PhD and their postdoc on something that is relevant to this particular story, they are going to be experts in terms of translating that to people who are not technical audiences because they're doing that every day as they're teaching fresh scientists and training that next generation. And then, of course, you can always come to us at SciLine and we will help you if you would like for free. We're philanthropically funded.
Can you tell us a little bit about what you have to offer at SciLine? This is a pretty great resource.
Yeah, so we are based at the American Association for the Advancement of Science. So those are the folks that publish Science, the research journal. We are editorially independent from that group, philanthropically funded. You can see all our sources on our website. And we do a couple of different things. The one that I've mentioned several times is our expert matching service. So that's when you come to us with your story and your deadline and you say, hey, I'm looking for an expert source. And our team of folks on staff who have a science background, mostly PhDs, a couple of master's degrees, we'll do all the legwork to find someone with the correct background and make sure they're available on your deadline, and then send that person to you and you do your interview independently. We also, for folks who may be in a broadcast realm, have opportunities for interviewing scientists in a broadcast setting. It's called Experts on Camera. Those are 2-hour blocks of time where you can sign up for a 15-minute interview. And we make sure that that scientist has sort of that comfort level to communicate in a broadcast format and has all the technology that they need to be able to do that and look and sound really good. And then we also do media briefings to help people get up to speed on the state of the research in a topic that's in the news.
It's a very helpful resource. The other thing that, it's kind of been drilled into me to always try to address is as much as possible cost. If you're talking about an early stage research, now, of course, one of its kind, even more so, like, could this be replicable, or putting in just like some cautionary language, right? Oftentimes takes a long time to reach a mass audience, can cost into the blah, blah. In this case, I think we had, as I recall, no idea what the real cost would be because this had been developed in such a unique partnership. But I'm just trying to think, before we open it up to questions, are there any other just like almost like a checklist of what are the points that you want to make sure that you're addressing that may or may not be specifically ironed out in a research article itself?
I think one thing that I don't think we've quite addressed yet is what were the precursors that came before this? Obviously, that's very hard to address fully in a breaking piece, but maybe that second day story or that follow-up. So what came before this will help you decide, is this something that's truly a first of its kind? We heard from Dr. Musunuru that they'd been working for 10 years to develop a technology that then enabled them to move really rapidly on this. So thinking about what came right before this. And then you mentioned cost, I think is really important. Another question that became addressed really well later on, but I think would have been good to address more fully right in the moment was, who else would this apply to? What other kinds of patients could get this type of a treatment?
The last thing I'll just say that I thought was kind of interesting with the initial round of coverage is, thinking a little bit about your audience when we are covering the quote unquote study story, right? The genre of research. Some of us might be writing for very specialized audiences, and we know who we're writing for in that context. Others of us are writing for more general audiences. And to some extent, in the digital age, I think we're writing less of the study story that's a national story and just like going to a local expert to speak about it, right? But there still is an opportunity here to reach new audiences and large audiences, but to also be really thoughtful about, like, are we defining what we need to define? So, at the Inquirer, I really pushed the reporter to make sure to explain what CRISPR is, right? It's very buzzy. Everyone's heard it. How many people can really explain what it is, right? This was an opportunity to do a little like light flourishing writing too, which is kind of fun, like that sort of like narrative, the backstory, and explain a complicated term in a way that'll make a reader feel smart. In our case, although our primary audience is always a Philadelphia reader, our story, got picked up in this case by Apple News, and I think that it was based on some of the numbers I saw, probably a pretty significant national, source of information about this, right? So at all points, we're kind of thinking about different audiences and what to make sure we're breaking down or not. I'd like to, at this point, see what questions you all have for our speakers. So why don't you, if anyone wants to jump to microphone, or what we do, it was 1, 2, and then we'll go from there.
Hi, and I apologize if this is something I should know, but with you talked about this particular kind of CRISPR base being able that you could use this with multiple liver conditions. Is there something about this that is specific to the liver, and you would need like a different platform for, gene dysfunctions in other organs, or is it just that the liver is a particularly good starting place? Thank you.
Yeah, that's actually a very astute question, and you're right on the mark. So the reason that the liver is open to us right now in a way that is not yet open for other organs is because the liver's job in life is to clean things out of the blood. So if you put a drug into the blood, in this case, the way we deliver CRISPR into the body is we make trillions of these little soap bubbles, and the CRISPR cargo is wrapped up in these soap bubbles. Intravenous infusion over about 24 hours, so straight into the bloodstream. If you put something into the bloodstream that way, it'll get picked up by the liver. The liver just grabs onto it and picks it up. And so that's advantageous if you're talking about diseases where if you're trying to make the fix in the liver, okay, great, because you don't have to do much extra work. Now, if you can get CRISPR into other organs, then that opens up whole different sets of diseases. And the potential is there. It's just not as easy to do. There you have to start changing the drug in certain ways. You have to change the chemistry, or you have to add, different doodads, if you will, onto the surface of the drugs that help to redirect them away from the liver into other organs, such as the muscles or the blood vessels or the blood stem cells and so forth. And so that's an area of very, very active research right now. Many people at the University of Pennsylvania, many people at other institutions are engaged in figuring out exactly how to change the drug, the basic CRISPR drug, in such a way that you can get it into other organs. And it will happen. It might take some time, but it will happen. It might take, a few years. It might take 10 years. It might take 20 years. But, I'm very excited because there's plenty of diseases, believe me, we can work on in the liver alone. We're talking hundreds of diseases. But every new organ that gets unlocked, as it gets unlocked, means hundreds more, thousands more diseases that will become amenable to treatment with CRISPR therapies, with gene editing therapies.
For the doctor, you may have just answered this, but how hard is it to change the GPS address? And then also, you talked about turning off the gene. I mean, could you turn on a gene or replace a gene?
Yeah, so it all depends on exactly what kind of change you need to make. But yeah, that's exactly right. So to change the GPS address is actually trivially easy. So in the laboratory, we can do it in like 24 hours. Now, if you're actually making a drug that has to go into a patient, into a human being, obviously, then you're starting to think about, okay, how do you manufacture things at a very high level, high quality standard and so forth. And so it's not quite as trivial, right? Now you got to make a whole new thing that has, like your new sequence, but it's still like making a new drug. And so that's why, instead of being able to do it in 6 weeks, it took us 6 months to actually be able to make the drug. But we think we can streamline that further. But conceptually, it's actually not hard at all to change that GPS. It's really what you can do with the technology depends on, what your goal is. So if you want to turn off a gene, what you can do with this technology is you can change a single letter in just the wrong place. I'd say right place, but in this case, the wrong place that basically flips the gene off. If you're starting with a broken gene that already has a misspelling, you can use the same technology to make that single letter change that corrects the misspelling to the right spelling, And that effectively is taking a broken gene and turning it back on. Now, if you want to do something a little more sophisticated, like put in a whole new replacement version of a gene, it's doable. It's just more complicated. Then it's not just the simple CRISPR system I told you about, where it's two components, where you have the editor and then you have the GPS. There you would need to add like a third component that actually has the template that has the new gene that you want to put into the DNA, right? So because you're adding some new element in, and so that needs to be a third element of the drug. And anytime you're adding, an extra component to the drug, that just increases the complexity substantially. And so we're starting to be able to do things like that, but it's not quite ready for prime time, I would say.
Thank you.
Hi, my name's Peter Werwine. I'm at a publication called Managed Healthcare Executive, and I'm an Inky reader. So I have two questions. I guess I just want to get this straight in my head. Forgive me. You're obviously not affecting just one gene and one gene edit in a single cell. So how many liver cells do you have to hit to have a response here? And are there some dose response dynamics in terms of how many genomes you're trying to edit with the therapy? And an unrelated question: gene therapy in UPenn, for those of us who are a little older, has a fraught history. The Jesse Geisinger case. So I'm wondering whether that came into your reporting and how that factored into your research at Penn.
Sure. So for the first question, so let me think here. So repeat the question. Just give me a prompt.
What you can end up with I think, a misleading cartoon that you are targeting a single cell and affecting one gene. And I don't think that can't be so. So I'm wondering how many liver cells you actually have to hit.
Yep. Yeah, and so it depends on the disease, right? So an advantage of urea cycle disorders, where you're talking about broken enzymes, is you don't need 100% working enzyme in order to handle something like ammonia. There's a lot of redundancy in the system. And so with most of these diseases, if you have even 50% working, you'll be healthy. It's the difference between what we call a recessive disease and a dominant disease. All of these diseases are recessive disease. So if you can get to 50%, that's more than enough because of that redundancy. You just have to have that much working enzyme. We do think for most of these urea cycle disorders, even as little as 10% might be enough. The challenge is it's hard to know exactly what the threshold is, and it might vary a little bit from person to person, depending on their individual unique characteristics. But we don't have to fix, to your point, we don't have to fix every single cell in the liver. If we can fix 10% of the cells, make the correction and say 10% of the cells, our sense is that that is good enough to take what is a life-threatening, devastating disease into a much milder, manageable one. So hopefully that answers your first question. And then the second question, again, if you can give me the prompt.
It was related to gene editing has a complicated history, particularly at Penn.
University of Pennsylvania. Well, I mean, we were mindful of it. It goes back, it was 1999 when Jesse Gelsinger passed. So that was 27 years ago. And, Becca and I were very cognizant that we were working in a group of diseases, urea cycle disorders. Well, guess what? Jesse Gelsinger had not the same urea cycle disorder, not CPS1 deficiency, but a related one, another one of the enzymes of that chain of 6 enzymes called OTC deficiency. And so it was very much on our mind. And, I can't say it didn't influence us. In some ways, we would have prioritized safety anyway. But I think, having that history in the place that we were made us just that much more motivated to do things safely. And so we didn't really get into it, over the last hour, but we actually ended up giving KJ 3 courses of the therapy. And the reason for that is we wanted to start with a very, very low dose, recognizing that no kid had ever received this kind of therapy. By that point, a bunch of adults had, but no kid, no infant had ever received this kind of therapy before. And so we wanted to go to a very low dose, almost a homeopathic dose, just to make sure it was safe. And the advantage of this kind of drug is you can actually give it more than one time. And so we gave that first very low dose just to make sure everything was safe, everything went well. And in fact, as you heard, it actually looked like even with that very low dose, it started to get some improvement. And then we came back a few weeks later and gave a slightly higher dose. So still not a high dose, but a slightly higher dose, with the idea that, okay, like let's see if he does even better. And all the signs were that he did do better after that second dose. And then several weeks after that, we came back with a third dose. So higher, but still not a high dose. Like I would say more like a middle dose, to sort of top things off. And then, my sense is that he had even more improvement from that third dose. And then at that point, we stopped because we start to see that, as we were bringing the dose up, there was some evidence of stress to the liver. Nothing concerning, nothing that wasn't expected. But we got the sense that if we went even higher with the dose, then we might just start to get into some issues. And so we said, he's improved. It's all about managing, the balance between benefit and risk. And so we said, okay, you know what? Gave him 3 shots at this. He's had a good response. Don't want to push our luck. Want to be responsible. And so we didn't go any further than that. And so that was our way of trying to manage the risk, in a situation where we recognize that, yes, there is the potential for harm here. You hope you avoid it, but anytime you're doing something entirely experimental that's never been done before, there is the potential for harm. That's part and parcel of doing experimental research, of doing clinical trials.
We have 3 minutes left and 3 people are standing in line for questions. So why don't we try to do a lightning round?
When the rare disease folks sometimes talk about gene therapy as like a cure, what are we seeing so far with these treatments? Is it like lessening the severity of disease? Are patients still going to have some kind of long-term care needs going forward? Or are they going to be pretty normal? Like, what does it look like? What is the reality so far?
Are we talking gene therapies in general?
Well, like in his case, you said he still needs to have like a reduced protein diet. Is that something that's going to require longer term, other types of hair?
I think the question is this a cure or
Yeah. So
what is the promise?
I have never ever used the word cure in this context or any other context.
Yeah.
And Becca and I have been very, 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 very clear and disciplined from the very first interview we did, when the story came out, that we did not envision this as a cure. The goal was to help KJ.
Mhm.
And we feel even now, it's early days. He has his whole life ahead of him, right? And we're hoping for the best. We're praying for the best. But who knows, right? There's no precedent for this. And we're very mindful of that. And so, from Becca's perspective, from my perspective, and Nicole can speak on her own perspective, our fondest hope in doing this was that we could take a very serious, life-threatening disease and make it into a milder, more manageable form of disease. We would have considered that even if all we were able to do at the end of the day was delay liver transplant by 6 months or a year, that would be a win because, as I said before, the younger you are when you get a liver transplant, it's much, much riskier. And so if you can get him to an older age before he needs a liver transplant, that would be a victory, considering that we're starting from zero. Like there was no precedent for anything along the lines of what we were doing before. So Becca and I are very pleased with, how things have turned out, but we would be the last people to ever call this a cure or that we saved KJ or if I have anything to share with all of you, it's that, just be mindful of not, indulging in irrational exuberance and using those types of terms. Because I do think it creates, unrealistic expectations for other patients and other families.
Thank you. We are at time. I don't see anyone rushing to come in, but as we're leaving, I'm going to switch to a screen that'll let you know how to reach out to folks with questions. Thank you so very much, Kiran, Nicole, Sara, and those of you who joined us today. Thank you.
Of course. My pleasure.
Thank you.